Bismillahirrahmanirrahim. Assalamu alaikum and good morning. So it's a pleasure for me to be here the second time presenting the Pakistan Society of Nuclear Medicine Conference. And also somebody was telling me yesterday, it's the first time the radiochemists are allowed to participate in the main session of the conference. So we are very thankful for the President of Pakistan Society of Nuclear Medicine, Dr. Mehdi Raza, and also the team for acknowledging us and for realizing or making us feel the part of this PSMA community. So I think everybody in this room understands what's FDG. So FDG is a glucose, it's a fluorine-18 labeled glucose analog, where one of the hydroxyl group of the glucose molecule is replaced with fluorine-18. FDG is the most versatile radio tracer used in PET imaging. In fact, I would say PET imaging is actually dependent on FDG. If there was no FDG, PET will not be that much explored as we see today. So we will try to understand some reasons why we are still FDG centric, why we have FDG dominance. And then we'll try to understand some of the limitations of FDG and where are the other tracers which are non-FDG tracers are performing better than the FDG itself. Then we'll also try to understand some of the challenges we have in tracer diversification, how we can adopt a multi-tracer facility, and what are the things we have to do to make it possible. So FDG, as I said, it's a central pillar in PET imaging. It's a fluorine-18 labeled analog. It's working? Okay, I can take the... Yeah, I can have it. Yeah, so it's an analog where one of the OH group of the FDG, the glucose molecule, is replaced with fluorine-18. This is the whole process of FDG synthesis. The process looks very simple, but there is a science behind each step. It starts with O18 bombardment with protons. So you bombard the O18 with protons to produce the F18. The F18, when it's produced, is surrounded by many water molecules. And it's not possible to use this directly to do this reaction. So what we have to do is we have to make this fluorine-18 naked or a good nucleophile so it can actually attack on this triplet and replace it. So how we do this, we have to use our anionic exchange cartridges, which we say the QMA cartridge. The whole solution, fluorine-18 surrounded by water molecule, is added to this, and it will retain the fluorine-18 and the water will go out for the recovery. The fluorine-18 is then taken out of this cartridge, and then we do the azeotropic drawing to make it a real nucleophile. That nucleophile is then added to this mannose triplet, which is the precursor of the synthesis, and then we do the acidic or basic hydrolysis to get the fluorine-18 or analog of glucose. So once we have this glucose analog, this is injected into the patient, and because the glucose has a property like it tells us about the metabolism, and cancer cells use this feature of the glucose. So in the cancer cells, there will be more metabolism, and more FDG will be going into the tumors, highlighting them like this. So when it's reached the point, they emit a positron, and as soon as the positron is emitted, it suddenly takes one of the electron to annihilate and emit two gamma rays in opposite direction. And this is the mechanism we use to get this FDG scan. So if we try to understand what are the FDG-centric reasons, so these are the 5 main reasons or categories we can say that are the responsible for FDG dominance. If you see the first one, which is the universal tracer with broad clinical applicability, this is the main reason FDG is dominant, because FDG use one property of the cancer cells, which is the glucose metabolism. That's even the normal cells, but the cancer cells have that property even better or even more. And this is what the FDG uses. to make it visible. The second thing FDG is, if you see all the PET radio tracers, I'm not talking about the metals at the moment, not the gallium, the copper, the zirconium. I'm talking about the carbon-11, fluorine-18, oxygen-15, nitrogen-13. So if you see those, FDG is the one with the longest half-life. So also there is a centralized radiopharmacy well established for FDG, which make it a universal tracer. The third thing is the clinical FDG has clinical proven guidelines. There are well-written, well-documented, well-accepted protocols for both imaging and for reporting the analysis. Then FDG has high demand, which ensures kind of economic stability. Like the other non-FDG tracers, it will be only applicable to one situation. But in FDG, it's applicable to many situations which secures the economic, or which makes sure the economic stability of it. And FDG is also known as a benchmarker now for all the tracers. Like we know the FAPI is going very well in the market at the moment, and all the FAPI tracers are being compared with FDG if we want to have it a good, or if we want to improve it further. Out of all these 5 reasons, there is one main reason also, which is the centric to FDG dominance, and that's the unavailability of non-FDG tracers. So many radio tracers have shown even better promise in disease detection than the FDG, but they are not produced here locally, and that's why the people are bound to use the FDG. So now I will give you some of the examples from the clinical work that will highlight the tracers which are performing better than FDG. So if you see some of the areas here, the neurodegeneration, the amyloid and tau imaging, which are known as the distinctive marker for Alzheimer's disease, we cannot detect them with the FDG. So we have to use the other possibilities like fluorobeta P, fluorotau C, or those tracers. Myocardial perfusion cannot be well explained with FDG, so we are dependent to have the other ammonia, oxygen-15, or rubidium for these studies. Dopaminergic systems, because we know the FDG, it only gives the cerebral metabolism, but it's not a distinctive marker for dopaminergic systems. So these systems cannot be mapped with FDG. Likewise, hypoxia imaging, cell proliferation, estrogenic receptors, PSMA, dotatate, all these tracers cannot be well explained with FDG. So one of this is actually the fluorobeta P molecule, and if you see, it's a very simple molecule which can easily be synthesized in the lab. But the problem is this molecule, I think the Eli Lilly company has rights on this molecule. So it's not easy to just reproduce it in the lab. But if you see these, it's a direct comparison between FDG and fluorobeta P. And if you see, this is the normal brain, and this is the disease brain, and FDG is almost, there are some distinctions you can point out, but FDG is almost kind of similar in both the situations. But if you compare this radioligand, here this is the normal brain, and here you can see a lot of amyloid plaque can be identified with this radiotracer. The second kind of area where FDG is not performing well is the detection of somatostatin receptors. We know that FDG is just give us the glucose metabolism, but the somatostatin receptors, they are heavily expressed on the surface of neuroendocrine tumors. So we need a specific tracers to evaluate them, not the FDG. And here if you see, this is a dotatate positive patient where we can see these kind of spots here. But these spots are not visible in FDG. So therefore, the non-FDG or the other tracers can even explain the situation or the staging or the spread of the disease better than FDG. Likewise, another patient where the dotatate negative, but FDG is positive here. So in order to assess the real kind of staging or the condition of the patient, we also need to have the non-FDG tracers with us. This is again a comparison between FDG and PSMA positive patient. And we can again see that there is this lesion or the spot available here with FDG, which is not present here. And likewise, there is not this spot in a different patient. It's not present here, and we can be seen with PSMA gallium. So many situations where FDG outperform, we have to have the other radio tracers with us. Now you may be thinking, okay, dotatate is going well, we know, but we don't have gallium. How are we going to do the dotatate labeling? Then in that case, we can use this strategy. So again, this is the dotatate peptide, but instead of here, it's actually an octreotide peptide, but instead of having the dotatate, we have the nota here. And nota can be labeled with aluminum fluoride. And this chemistry is very simple because this is actually the one radionuclide for all tracer approach. You can label the PSMA with fluorine-18, you can label the octreotide with fluorine-18, or this kind of thing. And it's also, this chemistry is aqueous. You don't have to dry fluorine-18 as well. So these are the kind of alternates for the places where we don't have the gallium, for example. And now you may be thinking, okay, let's make the aluminum fluoride, but is aluminum fluoride actually playing a role in clinical trials? Can we actually see it? It's as good as the gallium dotatate. So here is a direct comparison between aluminum fluoride octreotide peptide with a gallium dotatate. And you see that, and the same patient was scanned after 4 months here. And you see FDG actually pointed out a kind of node here, a line here, which was not in this dotatate. But this was observed after 4 months in the dotatate. So actually aluminum fluoride octreotide was able to identify this mark much earlier than the dotatate. So likewise, it's not only applied on prostate or neuroendocrine. For example, this is a FAPI ligand, and we can also see that this mark was identified, was not identified by FDG, but it's a bit better identified in aluminum nota fluoride. And the SUV here was only 2, and here it was a maximum SUV of 4. So these are other options where they have used the same strategy for the places where there is no gallium. Then the easiest way, for example, if you want to have access to the gallium, the easiest way is people use the gallium generator for this. But I'm against this strategy because the cost of this gallium generator is, I think it was at least 700,000 in reals, what we buy from in Saudi Arabia, so which is around 5 crore for one generator. So if you use the same strategy, but instead of buying it in the generator, if we produce in the cyclotron, that is a much better strategy. So there are 2 ways we can produce gallium-68 from the cyclotron. One is the liquid way. This is the liquid kind of setup where you have the target, you make a solution of zinc-68, either in HCl solution or either in nitric acid solution. You irradiate it in the cyclotron, then you do the bulk purification and radiosynthesis. This is the liquid setup. The problem with the liquid setup is we have limited yield. This setup is only applicable if you want to use the gallium for your center only. But if you want to supply it to the other centers as well, it's better to use the solid targetry system because the yield here is almost 10 times more than the liquid target. And that's the approach we use at King Faisal Hospital to do the electroplating, and then we do the target irradiation in the cyclotron, we dissolve the target, and bulk purification and radiosynthesis. Likewise, copper-64, copper-67 can be prepared as well using the liquid and the solid targets. So this is my last slide. So here I would try to highlight some of the issues why we are FDG-centric. The first reason is the limited applicability of non-FDG tracers. We all know that non-FDG tracers are applicable only for one situation, for example. For example, if you take FLT, if you take FDOPA, so they are only specific for one. But FDG is kind of the universal tracer. Then the second issue we have is the cyclotron dependency. So one thing is the fluorine-18 labeling. So everywhere when we get the cyclotron, everyone focus on fluorine-18. But the same cyclotron can be used for gallium-68, for copper-64 productions. And I would like to focus on this point. Please, you have actually instrument. Just use it properly to produce the gallium-68 and copper-64. It's only a little bit effort, but it can be done. The second thing, oh, actually I missed the third one here. Sorry, but the fourth thing here is the approval processes. So this is also a very kind of, because you know in radiopharmaceuticals, one thing is the pharmacy, the drug, and the other thing is the radionuclide. And in this case, the logistics or the regulatory things becomes even complicated. So if you want to have this whole setup smooth, harmonized, parallelized, it's very important. Everyone has to have some role in there. So the regulatory people, I will request them to have some kind of relaxations in the rules so we can actually grow better in the radiochemistry field. Then the logistics and supply chain is another issue. We know that buying the target material, having some sources is always complications from PNRA and those kind of things. So they also have to give us some kind of relaxation just to the better growth of radiopharmacy thing. Then limited collaboration between disciplines is also an issue, like you have the cyclotron, but people are not producing copper, gallium, zirconium, or other radionuclides. It's just because they are not communicating well, they are not updated with the recent literature or the system. So if we do these things, it's possible to do all that work here as well. And lack of trained manpower is also an issue, and we are happy that Pakistan Society of Nuclear Medicine is playing your role just to have a smooth or a better collaboration between the international experts and the local experts. So that was it from my side. I would like to thank the Pakistan Society of Nuclear Medicine for the invitation. And also, I would like to acknowledge all the institutes that have enabled me to be standing here in front of you people. So thank you so much for your attention.